today's AP Biology lesson. I'm Allison Kate, and oh no, that's not what's supposed to happen. Um, we're just going to get right to our warm-up today. So yesterday's lesson was on chromosome inheritance, and you were given a homework assignment that is a multiple choice question, and you can see you have a diagram, and it says a spermatocyte produces the following 4 sperm cells. These cells are the result of non-disjunction during which of the following phases? And so you can see that N represents haploid. So you've got 2 sperm cells that are N+1 and 2 sperm cells that are N-1. So your choices are A, interphase, either the G1 or G2, B, interphase, the S phase specifically, C, mitosis, D, meiosis 1, and E, meiosis 2. And since you have had many lessons on cell cycle and mitosis and also the process of meiosis, you can easily eliminate A, B, and C because we know that there is no haploid gamete production in any of those processes. So now we have to figure out, is it meiosis 1 or is it meiosis 2? So think about the events that happen and when either the homologous chromosomes are being separated or those chromatids are being separated. Okay, got your answer? All right, the correct answer is D. Meiosis 1 would produce this non-disjunction or the failure of those homologous chromosomes to separate. And as you can see, what results is a incorrect number of chromosomes in those gametes, and that is called an aneuploidy. You either have too many or too few in the resulting gametes. All right, so today's lesson is going to be on DNA and RNA structure. And so some of it will be a review from when we discussed nucleic acids earlier in the context of macromolecules. And then we're going to talk about DNA replication. We're also going to look at Science Practice 2BB, which is explaining relationships between different characteristics of biological concepts, processes, or models represented visually in applied contexts. So you can guess that we're going to look at the process of DNA replication, and we're going to look at some models that represent that process and being able to explain the relationships between the different characteristics involved in this process. You should be familiar with the kind of the concept or understanding of nucleotide structure, which was presented earlier, hydrogen bonding, which was also presented in an early lesson, and then the idea of 5' and 3' strands, which I'll review because that's very important to DNA replication. The terms double helix, leading strand, lagging strand, and Okazaki fragment will be helpful today. By the end of today's lesson, you'll be able to describe the structures involved in passing hereditary information from one generation to the next. You should be able to describe the characteristics of DNA that allow it to be used as the hereditary material. And you should be able to describe the mechanisms by which genetic information is copied for transmission between generations. So here's what you need to know. So first of all, just reviewing DNA and RNA structure and understanding that DNA and in some cases RNA is the primary source of heritable information. So in general, DNA is going to be double-stranded compared to single-stranded RNA. Now, there are many different types of RNA, and there are some that are double-stranded. There is only one type of DNA. When looking at the 5-carbon sugar in DNA, it's deoxyribose. And if you look carefully and you remember how we count our carbons, that orange highlight is showing you the functional group at carbon 2, and there is just a hydrogen atom. So it's without oxygen, which is where it gets its name, deoxy. And then if you look at the RNA sugar, at that same position, the second carbon, there's a hydroxyl functional group. So it has that O present. So it's the ribose. And that's where DNA and RNA actually get their names. And then there are some nitrogenous bases that differ in DNA and RNA. So in DNA, the nitrogenous bases are thymine, which is abbreviated T, cytosine, which is abbreviated C, adenine, abbreviated A, and guanine, which is abbreviated G. In RNA, thymine is not present, but a related nitrogenous base called uracil is present. So we have a U in RNA, and then we still have our C, our A, and our G present in RNA. So let's just review what a nucleotide is, and I'm showing you a DNA nucleotide in this case. There are three components. The first one is a phosphate group. The second one is a sugar. In the case of DNA, it's deoxyribose, and we have a nitrogenous base. And you can see that the phosphate group is bonded by a covalent bond. It's a phosphodiester covalent bond. And again, if you're looking at your carbons, here's the first carbon, the second carbon that I just talked about. There's our hydrogen, making it deoxy. Here's our third carbon, our fourth carbon, and the fifth carbon is actually above the ring of this pentose sugar. And so this is important to note that this phosphate group will either bond by a covalent bond to the 5' carbon or the 3' carbon. And I'll explain this again when we get to the replication part. And then our nitrogenous base bonds covalently to the carbon at the first position. So remember, a DNA nucleotide consists of a nitrogenous base, a 5-carbon deoxyribose sugar, and a phosphate group. Okay, so the 4 DNA nitrogenous bases are adenine and thymine and guanine and cytosine. I'll talk a little bit more about them in a minute, but in this image, I'd like you to notice that the nitrogenous bases are held together by hydrogen bonds. And in the adenine-thymine pairing, there are 2 hydrogen bonds, and in the guanine-cytosine pairing, there are 3 hydrogen bonds holding those 2 nitrogenous bases together. Okay, so DNA and RNA exhibit specific nucleotide base pairing that is conserved through evolution. Adenine pairs with thymine, so in DNA you're always going to see that AT pairing, and then in RNA you'll see the AU pairing. And then cytosine will always pair with guanine in both RNA and DNA. And again, there's a special name for guanine and adenine because they have a double ringed nitrogenous structure, and they're called purines. And cytosine, thymine, and uracil in RNA have a single ring structure, and they are called pyrimidines. Okay, and that's important to understand that you have a purine and a pyrimidine are always going to be pairing up. Okay, so adenine and guanine are the purines, thymine and cytosine are the pyrimidines in DNA, and then in RNA, uracil replaces thymine. So uracil is also a pyrimidine. Okay, so this image kind of gets back to that, how that phosphate group will bond to the 5-carbon sugar, and the way it bonds will exert a directionality on the DNA molecule. The 2 strands run in opposite directions, and we call this being anti-parallel, and it has really important implications for how the DNA molecule is replicated. So I'd like you to look at the left-handed strand, and you can see that we call this the 5' end because this phosphate group is first bound to the carbon at that 5th position, and then it alternates. So then 3, 5, 3, 5, 3, and so we'll end with an open 3' end. Same thing is true in the opposite or anti-parallel strand. In this case, the phosphate group is first bound to the open end is 3, then we go 5, 3, 5, 3, 5, 3, 5' end. Okay, so we'll get back to that when we talk about DNA replication. So one difference between prokaryotic organisms and eukaryotic organisms, and I know you know this because we've been talking about the difference, many differences in these organisms throughout our review lessons, but prokaryotic organisms typically have circular chromosomes, while eukaryotic organisms typically have multiple linear chromosomes. So I have an image for you. You can see the prokaryotic DNA, and then you can see the eukaryotic DNA. And then both prokaryotes and some eukaryotes contain plasmids, which are small extrachromosomal double-stranded circular DNA molecules. So you can see this would be the main genomic DNA, and then you get your plasmids, which again are double-stranded circular DNA molecules. I want to talk for a minute about the packing of eukaryotic DNA, and I'm going to come back and talk about this in detail on Thursday when we get into the regulation of gene expression, and I'll probably show you this slide again, but just to understand how all that DNA gets packed into our cells in the nucleus of eukaryotic DNA. It gets packed so that it's largely inaccessible to the machinery that's responsible for transcribing genetic information. So this is one of the ways that we regulate gene expression. Chromosomes are dynamic in structure, meaning that they're always changing. And you know that a little bit from the cell cycle and mitosis, where chromosomes are condensing and they're unwinding. So we see a condensed region, and it may be loosened or modified as needed for various cell processes. And in eukaryotes, the chromatin is packed using histone proteins. And those histone proteins, as we'll see on Thursday, can undergo chemical modifications that result in changes to how the chromatin is organized. So you can see we've got our double-stranded DNA molecule. Okay, and then the histone proteins, they have a tail and a head, and they kind of wrap around that to form what's called a nucleosome. And then those nucleosomes form loops, and then we can see that we have a chromatid that's got that chromatin all densely packed. And this is a replicated chromosome with two chromatids. And this is probably what you're most likely to imagine when you think about chromosomes, is that kind of X-shaped structure. Okay, this is the overview of DNA replication. These are kind of the important things that you can come back to and think about that are important to know about DNA replication. So first of all, DNA replication ensures the continuity of hereditary information. DNA is synthesized in the 5' to 3' direction, and I'm going to go over that in the next several slides. The replication is said to be semi-conservative. What that means is one of the strand of DNA serves as the template for a new strand, so that each strand is produced, one new strand is produced, and one is the original strand. In blue, I have the names of some enzymes that actually direct the process of DNA replication. So these are all various proteins that play a critical role in the steps of DNA replication. The first one is called helicase, and as its name implies, it will unwind the helix. So it unwinds the DNA strands. We next have an enzyme called topoisomerase, which relaxes the supercoiling of that DNA helix just in front of the replication fork. And I'll show you a picture of this in the next slide. DNA polymerase is the main enzyme, and the reason it's the main enzyme is it is what is going to add DNA nucleotides to synthesize the new strand. However, it requires a primer made out of RNA to initiate this DNA synthesis. There is an enzyme called primase that facilitates the construction of RNA primers. Then that DNA polymerase enzyme will come along and synthesize the new strands of DNA, and it will do it continuously in one strand on what is termed the leading strand, and then in segments or discontinuously on the lagging strand. Because the lagging strand is made in pieces, it takes longer, which is why it's termed the lagging strand. DNA ligase joins the fragments on the lagging strand, and the lagging strand fragments are called Okazaki fragments. So let's take a look first at the model of semi-conservative replication. And I will tell you, and I'm not going to go into it now, but there are elegant experiments that were done to show support for this model that is semi-conservative compared to completely conservative, or a model known as the dispersive model. So you can look at those experiments on your own. But we're going to start off with a parent molecule, and it's shown in dark blue. And then what we see, and what we're not being shown here, but the enzyme helicase is going to separate by breaking those hydrogen bonds between the nitrogenous base pairs, and it will separate those parental strands into templates. So they are the old strands that are serving as the templates for the synthesis of the new strands. So then we get the formation of those complementary new strands. So what that means is on this side, if the parent strand is T, then the new strand will be an A. If this is C, then we get G and T and A and T and A and so forth. So what you're seeing here is two identical strands result at the end of the replication process. So this is a model that predicts that when a double helix replicates, each daughter cell, which are shown in light blue here, will have one old strand, which is shown in dark blue, and one new strand, shown in light blue, that is derived from that parent molecule. Okay, so now I'd like to talk about the process of DNA replication, and I'm going to start with the leading strand. It's a little bit more straightforward, and what I'm looking at in this square is just kind of the large overview of how this process occurs, and we're actually going to get into the leading strand, which you can see here is shown in a continuous light blue arrow, and it's also shown in this way. So this is our replication fork, and these are the origins of replication. And so the overall direction of replication is, you can see the red arrows are indicating that. So first I want to focus on the leading strand. And again, here's our directionality. The template is this 5' 3', and so the new strand will always be synthesized in the 5' to 3' direction. You can think of this one as going toward that replication fork. Okay, so let's look at the details. First of all, the anti-parallel structure of the double helix affects replication. The DNA polymerases add nucleotides only to the free 3' end of the growing strand. So here's our growing strand. This is where we're going to be adding our new nucleotides. Okay, so a new strand can only elongate in the 5' to 3' direction. So pay attention to that arrow. DNA polymerases cannot initiate the synthesis of a polynucleotide. They can only add nucleotides to an already existing chain that is base paired with the template. And when we say base paired, we mean that they are connected by hydrogen bonds to the nucleotide, or the nucleotide, or nitrogenous base in that parental strand. So how do we get started then? The initial nucleotide strand is a short RNA primer. There's an enzyme called primase that starts the RNA chain from a single RNA nucleotide and then adds those RNA nucleotides one at a time, using the parental DNA as a template. So the red that you're seeing is actually the RNA primer. It's laid down by RNA primase, and it is going to bond those nucleotides in a complementary fashion. The primer is short. It's only between 5 and 10 nucleotides long. In case you're worried about it, it gets taken out later, and I'll show you how that happens. The new DNA strand will start from the 5' end of the RNA primer, right? So now our DNA polymerase can add to that existing RNA primer strand. And enzymes that are called DNA polymerases are what are actually going to catalyze the elongation of that new DNA at that replication fork. The rate of elongation is about 500 nucleotides per second in bacteria and about 50 nucleotides per second in human cells. Okay, so let's take a look at the lagging strand, and then we'll review the whole process. So again, here's kind of where we are. You are here, and now we're looking at these lagging strands. And so you can see them here, and you can see them here coming toward the origin of replication and away from the replication fork in both of those cases. So again, in this little picture, the light blue is going to be the newly replicated DNA, and the little red is our RNA. All right, I'd also like you to notice in this image that we have different pieces of DNA labeled 1 and 2, and these are the Okazaki fragments, which are the pieces that are going to be put together in discontinuous steps and pieces that will later be joined by an enzyme called ligase. Okay, so looking at how the lagging strand is actually put together, along one template strand of DNA, the DNA polymerase synthesizes a leading strand continuously, moving toward the replication fork. Okay, so that's what we talked about last time. Here it is, there's the fork, and here it is on the other strand, here's the fork. Okay, so to elongate the other strand, which we call the lagging strand, the DNA polymerase must work in a direction away from the replication fork. So can you see how it's moving away from the replication fork and toward the origin of replication? The lagging strand is synthesized in a series of segments that are called Okazaki fragments. After formation of Okazaki fragments, an enzyme called DNA polymerase 1, rather than the DNA polymerase 3, that is the main polymerase adding those nucleotides, DNA polymerase 1 will remove those RNA primers and replace the nucleotides with DNA, and the remaining gaps are joined by DNA ligase. So if you look carefully at the image, first in the first one, we see the RNA primase is this pink enzyme, and it is making the RNA primer, okay, in the 5' to 3' direction. Then we see DNA polymerase 3 will begin at the end, the 3' end of fragment 1, and it will synthesize the new DNA. And that is the Okazaki fragment. Then the DNA polymerase 3 will detach, and over here, DNA polymerase 3 is going to go back and start working on Okazaki fragment 2, okay? But RNA polymerase 1 is back here, and it will actually replace that RNA primer with DNA, and finally DNA ligase will form that bond between Okazaki fragment 1 and 2. So it's lagging strand because it takes a little bit longer to get all those steps done. So this is a summary. Okay, again, this kind of shows you where we are in the process. Here's our replication fork. It's not showing the other side. Okay, so again, call your attention. We've got Okazaki fragment 1, 2, 3, 4, and 5. We've got our helicase, which is the enzyme that unwinds. We have our single strand binding proteins, which I haven't talked about yet, but these are some additional proteins that once that replication fork is split, will kind of hold these together so that they're not kind of moving around too much. The topoisomerase would work up here as well. We have DNA polymerase 3, DNA polymerase 1, and DNA ligase. So a good thing to do is just kind of make sure you understand the actions of all of the enzymes involved in DNA replication. Okay, so what does helicase do? It unwinds the double helix. It unwinds that parental double helix. These single-stranded binding proteins stabilize the unwound template strands so that those enzymes can hop on and do their work. The leading strand is synthesized continuously in the 5' to 3' direction by DNA polymerase. Primase begins the synthesis of the RNA primer for that 5th Okazaki fragment. DNA polymerase 3 is completing the synthesis of fragment 4. So I've got those arrows for you to show you that. Where's my cursor? So I can show you that. When it reaches the primer on fragment 3, so here's the primer on Okazaki fragment 3, it will detach and begin adding DNA nucleotides to the 3' end of fragment 5 primer in the replication fork. DNA polymerase 1 will remove the primer from the 5' end of fragment 2, which you're seeing here, and we will replace it with those DNA nucleotides added one by one to the 3' end of fragment 3. After the last addition, the backbone is left with a free 3' end. DNA ligase finishes the job by joining the 3' end of fragment 2 with the 5' end of fragment 1. Okay, so this is actually, let me go back because that was pretty quick. So these are the enzymes and the steps, and this is kind of the money slide. So if you want to go back and study, this is the one to study. I left this image in here. This is the summary of DNA replication, but I have no words. And so what I thought you guys could do is to look at this and you tell the story. Okay, so you've got a lot of information in this model, and so you can paint a picture. Task verb would be describe the process of DNA replication based on this model. Okay, so one other thing, DNA replication is not without error, right? And so the DNA polymerase 3 does some proofreading and repair, and there are also some other enzymes that are involved in that proofreading and repair process. So DNA polymerases proofread the newly made DNA, and they replace any incorrect nucleotides. In a mismatched pair of DNA, other enzymes correct errors in that base pairing. One of those we'll see will be an enzyme called nuclease. DNA can be damaged by exposure to harmful chemical or physical agents, such as cigarette smoke or X-rays. These are environmental, physical or chemical agents, and it can also just undergo spontaneous changes or mutations. In nucleotide excision repair, a nuclease will cut out and replace the damaged stretches of DNA. So the error rate after proofreading repair is low, but it doesn't go to zero. So the DNA may have or likely will have some changes in their nucleotide sequence. The sequence change may become permanent and can be passed on to the next generation. But these changes, these mutations, are the source of the genetic variation upon which natural selection operates. So they're actually quite important. So let's get some practice with these concepts. The first one is a multiple choice practice, and it kind of is going to get at the idea of DNA base pairing. So there's another famous experiment that was done by a man named Erwin Chargaff that leads to Chargaff's rules, which are that A will pair with T and C will pair with did I say that right? A will pair with T and C will pair with G. So what he did is he investigated the nucleotide composition of DNA, and he analyzed DNA from various organisms and measured the relative amounts of adenine, guanine, cytosine, and thymine present in the DNA of each organism. And he did many more than the organisms that you see here. And so in this table, table 1, we see the relative amounts of bases by percent in an octopus, a chicken, a rat, a grasshopper, and a wheat plant. Okay, so you can study those percentages. Then we also have some percentages of C plus G and A plus T to give you some additional information. The question you're asked is, which of the following statements best explains the data set? Okay, so you're interpreting this data. A, since the percent of A and G add up to approximately 50% in each sample, adenine and guanine molecules must pair up in a double-stranded DNA molecule. Think about that. Do A and G pair up? Since A and T are approximately the same in each sample, adenine and thymine molecules must pair up in a double-stranded DNA molecule. So think about that. Do A and T pair up? C, since the percentage of A plus T is greater than the percentage of C plus G in each sample, DNA molecules must have a poly A tail at one end. We haven't talked about poly A tails. I will talk about them on Thursday. So at this point, if you don't know what a poly A tail is, that might be a difficult one to either select or omit. And then finally in D, since the percent of C and T add up to approximately 50% in each sample, cytosine and thymine molecules must both contain a single ring. So that's getting at purine pyrimidine. So hopefully you have your answer, and hopefully you selected B. So A and T pair up, A and G do not pair up, C and T do not pair up, and poly A tail is a distractor in this case. Okay, so one more, and this is related to prokaryotic DNA. So which of these following statements best explains the structure and importance of plasmids to prokaryotes? Plasmids are circular, single-stranded RNA molecules that transfer information from prokaryotic chromosome to ribosomes during protein synthesis. Are plasmids made of single-stranded RNA? Ask yourself. B, plasmids are circular double-stranded DNA molecules that provide genes that may aid in survival of the prokaryotic cell. So are plasmids double-stranded DNA? C, plasmids are single-stranded DNA molecules which are replicated from the prokaryotic chromosome that prevent viral reproduction within the prokaryotic cell. And D, plasmids are double-stranded RNA molecules that are transmitted by conjugation that enable other prokaryotic cells to acquire useful genes. So hopefully you remember from this lesson, because you were paying such close attention, that the correct answer is, of course, B. Okay, plasmids in prokaryotes are double-stranded DNA molecules. So you could immediately eliminate A and D since they're talking about RNA. And then you had to remember that it's double-stranded rather than single-stranded DNA. All right, so let's get some FRQ practice now. So this question says, scientists seeking to determine which molecule is responsible for the transmission of characteristics from one generation to the next knew that the molecule must, and there must be three features. One, copy itself precisely. Two, be stable but able to be changed. And three, be complex enough to determine the organism's phenotype. You are asked, your task verb is to explain how DNA meets each one of these three criteria stated above. This is a long question. Okay, so this was a long FRQ. This would be a 10-point question. It was an older question, so the scoring guidelines are a little bit different, but it is a 10-point question. So let's tackle the scoring guidelines. First of all, we just talked about copying. So the molecular properties for precise copying include the template concept. You could even talk about the replication being semi-conservative, that one of the strands is conserved and one is made brand new. You could talk about the molecular structure, where we have complementary base pairing, A pairs with T, C pairs with G. DNA polymerase is the enzyme that actually adds those complementary nucleotides to the existing parental strand. The idea that those strands have to separate, you could even talk about helicase being the enzyme that separates. And then any elaboration of replication, such as a proper sequence of steps, going into detail about leading and lagging strand, or discussing the enzymes involved in the process. The second part, you need to cover both stable and able. So what makes DNA stable, or the genetic material stable? Its shape. It's got a double helix. Okay. It's also stable because of its bonding. Remember that there's multiple hydrogen bonds between those nucleotides holding those strands together. There are silent errors. In other words, there may be some differences, but they're not always going to be in the part of the DNA that is expressed. And then finally, it's able to be repaired through that proofreading that we discussed at the end of our discussion about replication. And then able, right? It's able to change mutations, right? That replication process is not 100% faithful. There's other ways to inject change. We talked about crossing over during meiosis. We talk about mutations such as base substitutions, gene rearrangements. I know you discussed these yesterday, inversions and duplications and those kinds of things. And also DNA is sensitive to mutagens such as ultraviolet radiation or harmful chemicals in the environment. And then finally, how is it complex enough to determine the phenotype? We know that it's the nucleotide sequence of the gene that determines the amino acid sequence in a protein. And actually tomorrow, your lesson is going to go over exactly how that happens in the two steps in making a protein called transcription and translation. There are infinite base sequence combinations that can lead to the all of the different kinds of proteins that need to get constructed. And since proteins are responsible for the phenotype, there is infinite variety in the way that those proteins are produced. And then again, probably not today, but after you watch tomorrow's lesson, you could describe transcription and translation. So that was a long question. And if you wanted to try your hand at writing that, you should give yourself about 22 minutes. Okay, so a little bit more practice. This is a model of the molecules involved in DNA replication and their placement relative to each other. So you can see topoisomerase, you can see helicase, you can see RNA, and you can see the DNA double helix, and you can see the direction of those strands. One, the top strand, and the bottom strand have different directions. And you kind of have to look at the unwound portion to really get that. Okay, so figure 1 is a model, including molecules involved in DNA replication. Which of the following correctly explains where DNA replication will begin on the strand oriented in the 5' to 3' direction. We're reading from left to right. Okay. A, DNA replication will be randomly initiated along the unwound portion of the DNA strand since base pairing will occur. Okay, so you have to think, is there a random initiation sequence? B, DNA replication cannot occur since there is already RNA base pairing with the template strand. C, DNA replication will be initiated immediately to the left of the RNA since DNA polymerase requires an RNA primer. D, DNA replication will be initiated at the site of the topoisomerase since it is where DNA begins to uncoil. Okay, so that's just wrong on 2 counts. The helicase is where it begins to uncoil, creating the replication fork. So that one is just wrong. And we know that B is wrong because the RNA primer is necessary for the DNA polymerase enzyme to know where to start. So our correct answer, and I hope you guys got that, is C. All right, so let's do a quick check for understanding for our science skill. Again, we want to be able to explain relationships in the process of DNA replication in a model that's represented visually. So here you have a model that is showing you the process involving nucleic acids. They don't tell you what it is, but it's a model of a process involving nucleic acids. Which of the following best explains what process is represented in figure 1? A, new DNA strands are being synthesized in the 3' to 5' direction from their DNA templates. B, new DNA strands are being synthesized in the 5' to 3' direction from their DNA templates. C, the new RNA strand is being synthesized in the 5' to 3' end from its DNA template. And D, 2 new RNA strands are being synthesized in both directions from their DNA template. So I think you know that we can eliminate C and D, and now we need to understand our directionality. So is it going to be going in the 3' to 5' or the 5' to 3'? Hopefully you know that DNA is always going to be synthesized in the 5' to 3' direction because the new nucleotides are always added to the free 3' end. So here's your debrief. You should now be able to describe the structures involved in passing hereditary information from one generation to the next. You should be able to describe the characteristics of DNA that allow it to be used as the hereditary material. You should be able to describe the mechanisms by which genetic information is copied for transmission between generations, and you should be able to explain the relationships between different characteristics of biological concepts, processes, or models represented visually in applied contexts. And as has been our key takeaway for the last many lessons, heritable information provides for the continuity of life. All right, so let's take a look at a FRQ that was given on a recent AP Biology exam to look at some common errors and misunderstandings in this idea of DNA structure and replication. So first of all, figure 1 is showing you a COMET assay, which is an assay or test that's used to detect double-stranded breaks in DNA. Okay, before this question, nobody knew what a COMET assay was. And in fact, you don't really need to know because you're given all the information that you need in the question. So let's read it together. A COMET assay is a technique used to determine the amount of double-stranded breaks in DNA, which shows DNA damage in cells. The nucleus of an individual cell is placed on a microscope slide coated with an agarose gel. An electric current is applied to that gel, and that causes the DNA to move, and that process is called electrophoresis. And the DNA is stained with a fluorescent dye. When viewed using a microscope, undamaged DNA from the nucleus appears as a round shape, which is the head, and fragments of the damaged DNA extend out from the head, which is called the tail. The length of the tail corresponds to the amount of damage in the DNA. And I'd like to point out one more thing. In this diagram, you've got a negative side and a positive side. Part A, To explain the movement of DNA fragments in the COMET assay, identify one property of DNA and provide reasoning to support how the property contributes to the movement during the COMET assay technique. And then in part B, in a different experiment, cells are treated with a chemical mutagen that causes only nucleotide substitutions in DNA. Okay, remember what the COMET assay detects. It detects double-stranded breaks in DNA. It asks you to predict the likely results of a COMET assay for this treatment. Okay, so let's look at the scoring guideline for part A. You needed to identify that DNA has a negative charge, which means that when you place that electric current on it, it will move toward the positive pole. The other thing that needed to be identified is that DNA can be of different sizes, and we know that different sizes of DNA fragments will move at different rates in an agarose gel. The smaller ones moving more quickly because the gel is porous, and the small fragments can travel through the pores at a different rate, at a faster rate, and the large fragments have more difficult time getting through those pores. Okay, so that part was worth 2 points, and you needed to get both the identification and the reasoning to be able to get those points. And then you needed to make a prediction about what the likely results of a COMET assay would be if you treated it with a chemical mutagen that simply causes a substitution. And again, you would have a prediction that there would be a head only or a head with no tail. And then the tail will be shorter than with a double-stranded break in DNA. So you could have said either of those predictions. Okay, so let's read a student response. That was a perfect 3-point response. DNA is negative. Therefore, when applying an electric current, DNA will move towards a positive charge. Smaller pieces of DNA move through the agarose gel more easily than larger pieces, which is why the smaller damaged fragments of DNA are drawn out more towards the positive charge or the tail. And in part B, all of the DNA will remain in the head, the nucleus, because with only nucleotide substitutions, there won't be any damaged fragments to travel outside. So what were some common misconceptions or knowledge gaps? So in part A, students often identified DNA as either a positive charge, oops, or uncharged, oops, or polar, or has both negative and positive ends. So there was general misunderstanding of the idea that DNA is negatively charged. Some students thought that the charge of DNA depended on whether it was damaged or not, and that negatively charged molecules were attracted to the negatively charged end of the gel. So be straight on DNA is negatively charged due to all those phosphate groups, and it will move toward the opposite charge, which is positive. So a response that demonstrates the understanding of the charge of DNA. DNA is negatively charged, therefore, during electrophoresis, it moves toward the positive end of the gel. And then in part B, many students were unsuccessful at predicting the results of that COMET assay. Some students predicted that the results of the assay would be the same or they would be different, but they did not give a comparison. Okay, so that's just remembering to complete your thought. A misconception commonly seen was the lack of a tail would mean the COMET assay is inconclusive. Okay, so a common misconception or gap in knowledge was that there were incorrect predictions, and a response that demonstrates understanding is there will be a head and no tail since the DNA isn't broken in nucleotide substitution. All right, so here's your homework. This is your homework for tomorrow. You'll be with Ms. Pfizer tomorrow, so she'll review this with you. And tomorrow's lesson will be on protein synthesis. So again, this is looking at 4 samples of nucleotide distribution in 4 nucleic acid samples. And the researchers were studying new viruses. They analyzed genetic material found in 4 different virus samples to determine the percent nitrogen-based composition of each virus. The data are shown in the table. Your question is, which of the following samples most likely contains double-stranded RNA? Okay, so this is a little bit different than we've been talking about. So taking the information that we have learned in this lesson and applying it to a new situation. Okay, so I wanted to end today by giving you a little bit more information about some additional resources that you can find on AP Central. The first thing you need to do is log in at myap.collegeboard.org. Use your username and password to sign in. Oh, by the way, if you don't have an account, you can create an account. Then you're going to scroll over to where it says AP Classroom and click there. And also notice that it tells you when your exam day is and if you've registered for the exam or not. Then there's a little bit more information here. You can click here. This is where you can see your AP Live class that you're watching right now. And then this is where you can get some practice answering free response exam questions. So click there for optional practice questions. And what you'll see is you'll see all of these are unlocked for you. And you can look at what unit, and then what the free response question is going to be going over. Is it concept analysis? Is it interpreting and evaluating experimental results? So you can look at what unit and what skill you want to practice, and then you click begin, okay? Then you go ahead and answer your question. You can actually write in your response in the place provided. And then you're gonna get a prompt that's gonna say, are you sure you wanna submit your assessment? Click yes. And then once you click yes that you wanna submit, you'll get a box and it will ask you if you wanna review your the responses, the scoring guidelines basically for this question on the AP exam. So if you want to review, check it out and then revise your answer, you can click yes. And if you're good with your answer, you know you already got the best score you can get, then click no. And then what you'll see if you click yes is you'll see examples of what a response should look like to earn the maximum number of points. That's super useful. And then you can see how it's scored on the AP exam. So click yes, review, or oops, no, submit now. Okay. So you have a to-do list, and your to-do list is make sure that you have your device and internet connection all ready to go. Check to make sure your email address and phone number are in your AP account, and tell your teacher if you are testing. Thanks for watching. We hope you'll join us again. This is the last week of our review lessons, so make sure you do your practice question for homework. Ms. Pfizer will review it with you tomorrow when she gives her lesson on protein synthesis. So thanks for watching, and I'll see you again on Thursday.